So I'm just going to introduce our project, which is aiming to develop antivirals against alphaviruses. So it's really a very interactive team. So I'm at Albert Einstein. A person not talking today is Andres Meritz, who's a RNA virus replication person, especially for alphaviruses. And then you'll hear from Tem Morrison. Tim, I can't call you Thomas, sorry, I'm just not used to it. Okay, and we've really benefited from all the folks who are part of the cores in our U19. So I'm basically just listing the papers here. And this is really people from across the U19. So that's been really a lot of fun. Okay, so just a quick overview about alphaviruses and their diseases in humans. So they're transmitted by mosquito vectors in general. They're arboviruses. There's a group that causes, they're called arthrodogenic. So that includes chikungunya, Mayoral, Anyanyong, and Ross River virus. So the symptoms are severe joint pain and inflammation, and importantly, they can cause a debilitating arthritis that can last for years. So we're going to really focus on chick today. There's also an encephalitic group that, like Eastern equine encephalitis virus, for example, those tend to have high mortality rates and cause encephalitis and have severe neurological sequelae. Okay, so I just introduced here the alphavirus RNA replication machinery. So it's these 4 non-structural proteins. The NSP1 really makes the whole complex associate with the membrane. It starts out on the plasma membrane and then can get internalized. And then NSP4, what we're going to hear a lot about is the RDRP, the RNA-dependent RNA polymerase. And our projects really have focused on identifying inhibitors of NSP4. I won't talk about these other aspects, but then Tem is going to tell you all about the in vivo efficacy studies. So today we're going to really focus on a compound that was originally identified and developed by the Emory Institute for Drug Development. So rather than calling it 2749, I'll just call it 4-fluorouridine. So it's a ribonucleoside analog. Okay, so 4-fluorouridine was in studies from our lab was found to be a potent inhibitor of alphaviruses. So we tried a bunch of different alphaviruses, and you can see that it had a high inhibitory efficacy, less so for Synvis and some like forest virus. So then just to summarize, this is a lot of work that was in the first paper. We saw strong inhibition of virus production for chikungunya, for a reporter virus that expressed luciferase, and in replicon assays. So it was clearly a replication inhibitor, not entry or other things. Time of addition studies showed that the first 4 hours post-infection was critical. And virus breadth showed that it worked against all those alphaviruses I show you, with somewhat less robust inhibition of SFB and Synvis. So then from Andres's lab, they did transreplicase assays with mixes of different components and really showed that the sensitivity correlated with NSP4. So it was clearly targeting the RDRP. Okay. So then Pei-Shi Yin in my lab set out to really select for and characterize mutants that would have decreased sensitivity to 4-fluorouridine. So what he did was take the attenuated chick strain 18125 and serially passage it on U2S cells in the presence of fluorouridine. And so he did 6 cycles of that, and at each cycle he monitored for the growth in the presence of the drug. And then at the end, we performed through the genome core, whole genome sequencing of the pooled virus and also of plaque purified viruses. So after all of that, I'll just show you how it looks. So here's passage 1. You can see that there's something like a 2-log decrease for all of the drug treated samples. These are the DMSO controls. And by passage 6, you can see that all of the selected samples, this is just pooled virus now, look resistant. And so these were all sequenced, and this was really the core D, which was Alex Greninger's group, and they were like fantastic to work with, I have to say. Okay, So what did this show? So we found that the sequence analysis showed selection for pairs of mutants, and they were on either NSP4, a Q192L that came along with an NSP2 mutation, H687P, or a mutation in NSP4 cysteine 483 to tyrosine, and that came along with another NSP2 mutation. So then Peishi engineered all of these mutations into infectious clones of the 181.25 strain or the parental strain. That's the clinical isolate. And what he found was that really it was the NSP4 mutation in each case that conferred reduced sensitivity. And you can see that when we say reduced sensitivity, it's not like it's 50-fold reduced. It's like at 4 or 5-fold reduction. Then Andres's lab also performed NSP4 replicase and transcription studies and found that the NSP4 mutations in these in vitro assays also conferred reduced sensitivity. And notably, all the mutants, all of the mutant viruses and remain sensitive to higher concentrations of fluorouridine. Okay, so where are they located in the replication complex? So they're on NSP4, as I told you. So here's a little schematic of NSP4. Has the typical, finger, palm, thumb domain arrangement. And you can see that Q192L is located here. The cysteine 483Y is located here, relatively close to the active site. So then this is a cryo-EM structure that was developed. And you can see that there's the NSP1 that's hanging onto the membrane. And then sort of in the pore is found the NSP4 protein, and it's associated with NSP2, although the NSP2 it only partially was solved in this structure. And then here's the NSP4 alone in a crystal structure, and you can see that here's the active site and where the NTP would enter. And here's the cysteine mutant and the Q192 mutant. So those two NSP4 residues are conserved across CHIK, clinical isolates, et cetera, and all the alphaviruses we looked at, whereas the two NSP2 mutations, which really didn't boost resistance appreciably, were in the methyltransferase-like domain. So that's not conserved among alphaviruses, and it's not shown in the structure, so we don't really know how it might be affecting the NSP4 mutants. Okay, so then Peishi performed some fitness studies. So basically mixing the mutant with the wild type and then passaging it on either U2S cells, here shown for both mutants, or on mosquito cell line C636 cells. So you can see basically the wild type outcompetes the Q192L mutant, which definitely has a fitness cost, and doesn't really outcompete basically the C483Y mutant is more or less equivalent. And I'm just going to show one result from Tem's lab here, but he'll tell you a lot more about all of this. So if you mix the two viruses, again, like we did here, wild type and mutant, and you inject them into the foot pad of a mouse, you can see that even in the same ankle there where the foot pad was injected, there's Q192L doesn't do so well against wild type, but in the dissemination where you look at the contralateral ankle, both viruses are basically either equivalent to wild type or outcompeted by wild type. Okay. And then an important point was, do these mutations change the fidelity of NSP4? So here we collaborated again with Alex Greninger's group in Core D. What Peishi did was actually generate from infectious clones a P0 stock, and then it was passed once on either U2S cells or C636 cells. And then we sent everything to Alex's lab, who did the sequencing. So when they did that, what they found was that the mutations did not really change the mutational frequency or have a propensity for increasing mutations in specific sites. That was important because earlier studies, which actually had looked at the C4083Y mutant in CHIK, thought that there were fidelity changes, but we didn't find that, and neither did subsequent publications. Okay. So I'm going to stop there, and Tim can now share his screen. Okay. And we don't hear you if you're saying anything.
Yeah, I think you're muted.
Yeah, sorry about that.
Oh, that's okay.
Go back to sharing my screen. Does that look good for everybody?
Yeah.
Okay. For this section, then, I just wanted to start with some additional background on the disease chikungunya. So the translation of chikungunya and the related anyang really illustrate, I think, the outcome of infection. So chikungunya translates as that which bends up, and the related anyang translates as painful weakening of the joints. And the series of chikungunya virus outbreaks that we've witnessed over the last 20 years that have involved millions of patients have really given us a better understanding of the clinical manifestations of infection. And so from this, 3 clinical stages have been defined. There's an acute stage that typically lasts 1 to 3 weeks. As Margaret mentioned, it's characterized by kind of a rapid onset of high fever and intense pain and inflammation in joints. This typically occurs in peripheral joints, such as the wrists and ankles, and joints in the hands and feet. And that's illustrated over here in this schematic. So there's typically a large number of joints involved, and kind of not illustrated here is it's also typically symmetrical. And then there's a post-acute stage that lasts from 3 weeks to 3 months. It's characterized by the persistence of this joint pain, arthritis, and tenosynovitis. And this occurs in greater than 50% of patients. And then there's a chronic stage that lasts for greater than 3 months. It's very similar to this post-acute stage in terms of this persistence of joint pain and inflammation. And a recent meta-analysis of a large number of published studies came to the conclusion that this occurs in approximately 51% of patients. And these images on the right were sent to me by a rheumatologist working in Brazil and just illustrate this chronic arthritis that can be observed in patients experiencing chronic chickpea disease. So a little more background on this chronic form of the disease. There's been a number of risk factors that have been identified. This includes female sex, age over 45, the magnitude and duration of viremia during the acute phase, and the severity of the disease signs and symptoms during this acute phase. In addition, differences in the anti-chickpea antibody response have also been associated with either a resolution or progression to chronic disease. And that includes this rise of early neutralizing activity as being shown to protect against chronic disease, suggesting then that antiviral immune responses are important, and controlling clearance of the infection are important in controlling whether someone progresses to chronic disease. The mechanisms of this are really not very well understood. There's a number of different hypotheses. One includes the induction of an autoimmune response. However, many patients with chronic chickpea disease do not show any signs of an ongoing autoimmune reaction. Another idea is that there's kind of unresolved injury in the joint tissue. So tissue damage during the acute phase or exacerbation of underlying damage during the acute phase may lead to these chronic symptoms. There's some evidence for that because underlying joint injury actually also is another risk factor for chronic chickpea disease. And then another idea is that there's long-term viral persistence or the persistence of viral PAMPs or antigen in tissues. And so there's some limited evidence of this in humans. So viral antigen and viral nucleic acid have been detected in a very small number of samples collected from human patients. There's stronger evidence for this in animal models. That includes both mice and non-human primates. And I'll talk a little bit about that in a minute. So kind of with all of that in mind, we and others have developed mouse models that recapitulate at least some of the aspects of this acute chickpea disease. So that's illustrated here. So in this model, wild-type C57 black 6 mice are inoculated in the foot pad with chikungunya virus or related arthritogenic alphavirus. And in the chickpea model, these mice develop this kind of characteristic biphasic swelling in the inoculated foot and ankle. So that's shown here. There's an initial peak at around day 3 post-infection with some subsiding, and then another peak around day 6 to 7 post-infection, and then an eventual resolution over the next couple of weeks. In addition, these mice develop histopathological changes in musculoskeletal tissues. That includes arthritis, tenosynovitis, and myositis. And as illustrated here, the mice also develop some disorganization of lymphoid tissue. So this is an example of a lymph node from a chickpea-infected mouse at day 5 post-inoculation, just illustrating that there's widespread kind of disruption of normal organization. So in addition to those kind of histopathological changes, we can also measure virological features. So the mice develop viremia, that's shown here. We can measure that. And we can also measure viral burden in tissues, predominantly musculoskeletal tissues, as Margaret kind of showed earlier. So showing here this ipsilateral ankle, which is proximal to the site of inoculation, but there is dissemination of the virus, and we can measure viral burden at distal sites. And I'll just note that there are mouse strains that develop more severe musculoskeletal tissue injury. And there are similar models that have been developed with other alphaviruses, including myarovirus and Ross River virus. So all of these can be used to evaluate new medical countermeasures such as antivirals. And then in addition to that, we and others have made efforts to develop animal models that mimics at least some aspects of the chronic form of the disease. And so some of that's shown here. So what we have found, and this has been reported by others, is that there's long-term persistence of viral RNA in joint-associated tissues of wild-type mice. So that's shown here. So that occurs in both tissues that are proximal and distal to the site of inoculation. And this long-term persistence can really be detected for weeks and months after initial inoculation. And this is observed in mice that are inoculated with wild-type strains of the virus, and it's not observed in mice that are inoculated with an attenuated strain, such as this 18125 strain of chikungunya virus, which is shown here in red, and this is successfully cleared from these joint-associated tissues by about 28 days post-inoculation. So this long-term presence of viral RNA is really a feature of wild-type strains of the virus. And then importantly, this persistence of viral RNA in these tissues is associated with histopathological changes, including synovitis and tenosynovitis. And so this model is interesting because we can use it to test whether intervening during the acute phase could then prevent this persistent infection and long-term histopathological changes. And we can also use this model to ask whether or not when we intervene at these later stages, would that treatment diminish the levels of this persistent viral RNA and then also impact the severity of these inflammation and injury in these tissues if we intervene later? Okay, so in these studies then, to begin to evaluate the in vivo efficacy of this 4' FLU that Margaret introduced, we performed the following study. So again, we have these wild-type mice. They're inoculated in the foot pad with wild-type chikungunya virus. And then at 2 hours post-inoculation, we initiated treatment with 4' FLU or vehicle only. So this was done by oral gavage 1 time per day and the dose of 5 mg per kg. And then we measured the swelling phenotype that I described, and then we also measured viral burden at different time points throughout the course of infection. So shown on the right here, treatment with 4' FLU did diminish the swelling that was observed in these animals, in particular this kind of later stage of swelling that occurs around 6 to 7 days post-inoculation. However, I'll note here that at least in this particular study, it did not eliminate this virus-induced swelling. And then, as I mentioned, we measured viral burden. And so shown on the left here is measuring viral burden in tissues at just 1 day post-inoculation. So alphaviruses are very fast replicating viruses. So we wanted to measure viral burdens at early times post-inoculation. So at this 1 day time point, I'll note that the mice at this stage had only received a single treatment of the 4' FLU. Nevertheless, we did detect decreased viral burdens in the ipsilateral ankle, which is again very close to the site of inoculation, as well as a greatly reduced viremia at this time point. And then by 3 days post-inoculation, we did detect reduced viral burdens in the tissues both proximal and distal to the site of inoculation. Okay, and then finally, we measured viral burden at 5 days post-inoculation. Those prior studies, we were measuring infectious virus. Here, we're measuring viral RNA because of these later times. It's actually more accurate to use a viral RNA analysis. And again, we detected substantial reductions in viral burden in tissues, particularly these joint-associated tissues that are distal to the site of inoculation. So that's shown here, this contralateral ankle, ipsilateral wrist, and contralateral wrist. So these data were very promising, we thought, because it does show really strong diminishments in viral burdens in tissues during those first 5 days of infection. And I'll note that we also performed some additional studies in mice to try to determine the lowest efficacious dose. And those studies revealed that a dose of between 1.5 to 3 mg per kg was the most effective and very similar to this 5 mg per kg dose that we're using in these studies. Okay, we also wanted to evaluate whether or not this compound had efficacy against a related alphavirus in mice. And so we used a very similar model. So here the mice were inoculated in the foot pad with myrovirus, which is an arthritogenic alphavirus found in the Americas. And again, we initiated treatment at 2 hours post-virus inoculation. And again, this was done once per day and by oral gavage. And as I'm showing you here, I'm showing you viral tissue burdens at 3 days post-inoculation. And similar to the chikungunya virus studies, we find that mice treated with 4' FLU had greatly reduced viral burdens in a variety of different tissues compared with the vehicle-treated mice, indicating then that this compound has in vivo efficacy not only against chikungunya virus, but related alphaviruses, which was consistent with the studies done in Margaret's group in the cell culture experiments. Okay, so then we also wanted to try to do studies to establish the treatment window, at least during the acute phase of infection. So we used a very similar experimental setup. Again, the mice are being treated once per day with 5 mg per kg by oral gavage. And so in this case, we initiated treatment at 2 hours post-inoculation. And then we had some additional experimental groups where we initiated treatment at 24, 48, or 72 hours post-virus inoculation. And in each case, we performed these swelling measurements. And in these particular experiments, when we initiated treatment at 2 hours post-inoculation, this greatly abrogated this swelling that's observed in virus-infected mice. And when we initiated treatment at 24 hours post-inoculation, we detected some modest effects on virus-induced swelling. And then by 48 or 72 hours post-inoculation, if we delay treatment to those time points, we did not detect any impact on this virus-induced swelling. And so this experiment then was terminated at day 10 post-inoculation, and we collected tissues for viral burden analysis. And in this ipsilateral ankle, which again is proximal to the site of inoculation, we only detected a significant difference in viral burden when we initiated treatment at 2 hours post-inoculation. However, at these distal sites, we did detect significant reductions in viral burden when treatment was initiated at 24 hours post-inoculation or at even later time points, suggesting then that this treatment can have at least some efficacy when the initiation of treatment is delayed relative to the time of infection. Okay, and then finally, kind of building on some of the information that Margaret presented back to these mutations that confer reduced sensitivity to 4' FLU, the Q192L and C483Y mutations in NSP4. We wanted to test if those mutations had any impact on the ability of the virus to replicate and spread in mice. And so viral burden analysis is shown here. And what we found in these various tissues, both proximal and distal to the site of inoculation, is that the introduction of the C483Y mutation had really no detectable impact on viral burdens. They're essentially identical to viral burdens detected in mice infected with the wild-type virus. However, mice infected with this virus containing the Q192L mutation showed greatly reduced viral burden in tissues, consistent with the competition studies that Margaret showed, indicating that this virus is, for whatever reason, highly attenuated in mice. But importantly, the other mutation does not confer any notable advantage in mice. So to just summarize what we've presented today, what I just showed you is that 4' FLU has in vivo efficacy in mouse models, and this is against both chikungunya virus and myrovirus. And importantly, this occurs when we deliver the compound orally. Margaret showed that selection with the compound identified mutations in NSP4 that decrease the sensitivity of the virus to the compound. And NSP4 is involved in both transcription and replication of viral RNA. And again, these mutations decrease the sensitivity to inhibition of those activities. But we think importantly, these mutations show there was really no change in fidelity of the RDRP of the NSP4 protein. There was no increase in virus growth in a variety of different contexts, including mammalian and mosquito cells or in intact mice. And similarly, there was no fitness advantage in these different contexts, and we did not detect that there was any alteration in any concerning alteration in chickpea pathogenesis. If anything, it was very similar or attenuated. So we think those are really important data because it suggests that this compound, while being an effective antiviral, does not select for mutations that have concerning impacts on properties of chikungunya virus. Okay, and so with that, I think that concludes our presentation. Margaret kind of thanked everybody at the beginning, and I'll just reiterate that this has been a really outstanding and collaborative project. So not only within our groups and Margaret and Andres Merritt's groups, but with the greater AViDD project. So all of the core facilities have contributed greatly to this. And so just want to thank everybody for their time.